Hi all, my name is Pablo Fernandez from the University of Campinas in Brazil, and I am here to explain today how we couple a sample collector to an electrochemical cell at the Campinas Electrochemistry Group. As you probably know, this is not our idea. This method was published in 2020 by the group of Professor Marc Koper at Leiden University. So I need to start this explanation by thanking to Professor Marc Koper, of course, but also to Youngkook Kwon, who was the other author of this contribution. Another person to thank is to Yurat Birja. It's extremely difficult to pronounce the name, and who was actively working with the technique in 2015 in Leiden when I was there. He was a great colleague and taught me many things about the equipment and how to set this experiment. Therefore, when I came back to Brazil in 2016, I was able to install the equipment in my laboratory, and this is what I am going to explain in this video. So, this is the equipment that we have. You can see a pump here. I will show in some minutes what we have in the air, and here you have the module just to connect the equipment with the computer. I mean with the software to control this equipment. Here the equipment is open. You can see a robotic arm here and many glass tubes that you can see better here, the glass tube and the robotic arm, where we can deposit the different samples that we take. Okay. Here we have an enlarged image of the sample collector, and here you have a sample compartment with the liquid, so you can pump the liquid through this PEEK tube. It can be also the Teflon, but in this case it's a PEEK tube, and it is connected with the robotic arms that I show in details before. So this is a quite flexible method, and you can pump liquids to different rates and to deposit different quantities or different volumes in the glass tubes. So to perform an electrochemical experiment, we use an electrochemical cell as the sample compartment. Here you can see a working electrode. In this case, it's probably a single crystal of platinum in an electrochemical cell. You can also see the electrolyte, and different from a conventional experiment, you can see the PEEK tip that I showed you before. The tip is deep inside the electrochemical cell, and you can pump the liquid from this part of the cell in this case. Well, the point is that in general we do not want to pump the electrolyte from the bulk of the cell, at least in our group, and we want to get what is produced close to the electrode. Then you just have to approximate the PEEK tip to the face of the electrode. In this case, we have again a metallic electrode, but you can use any other electrode you want. For example, it can be an FTO electrode with a semiconductor, and you can perform the fraction collection experiment in the presence or in absence of light, for example. Okay. We have published several papers. I will include some links in the description of the video, but I would like to finish with some recent result. Here you can see a cyclic voltammetry experiment for the electrooxidation of glycerol in alkaline medium. Please do not pay attention neither to the result in the presence of light or to the backward scan. Okay, the important thing here is to see the current potential profile. Here we can see a clear oxidation of glycerol, and by performing the fraction collection, we want to find out which are the products formed during the electrochemical potential scan. Therefore, what we did was, at the same time that we scanned the electrochemical potential and measured the current, we took samples and then we analyzed the samples by using HPLC and determine the concentration of several products. So you can see here how the concentration of different products change at the same time that you can measure how the current change with the electrochemical potential. So let's summarize how it works. Here you can see again the electrochemical cell, and here in more detail, in this case a glassy carbon electrode with nanoparticles on its surface, and we can see close to the electrode the peak tip, and here you can see the peak tube, and here the tube leaving the electrochemical cell. So what is generated at the surface is pumped out of the cell all the way through to the robotic arms where we collect the sample. So, as I said before, afterwards you will collect the sample in different tubes and you can analyze what you have inside these tubes, typically by HPLC or NMR, but you can use another technique. Well, that's all. Hopefully it is useful for some of you. Thanks for watching.